All right, hello everybody. We're going to go ahead and get started today. Today we're going to be looking at the cell cycle in terms of the M phase and interphase and how cells reproduce to make copies of themselves. So one of the responses that cells can do after they receive signals is to be able to divide and grow. And to do this, cells are going to have to go through two highly regulated processes. They're going to have to go through the interphase, in which the cell either rests or prepares for division, and then the M phase, where the division actually occurs. Now, most of the cell's life is going to be in this part here, this interphase. This is about 90% of its life, and only when it absolutely has to divide will it do this last 10% of the M phase. So we're going to go over the M phase today. We're going to go kind of break down these steps that break up the M phase piece by piece. A lot of these should seem familiar. You've definitely learned them in the past, but we're going to go over them again here. So the first step of the M phase is the prophase. That's where the cell is going to go through a couple processes to get ready to split. First, the DNA is going to condense into chromosomes. So you can see the free-floating DNA in the nucleus. This is typically how it exists. However, when it's getting ready to divide, it needs to be able to put into a shape that can be split in half to ensure that both daughter cells are getting all the DNA that they need. So the DNA is going to condense into these chromosomes that we see here. Next, the nucleolus, which is the membrane around the nucleus, is slowly going to start to disappear as we need to interact with these chromosomes. And then the meiotic spindles will begin to form. So we have these spindles coming out from these centromeres, and they're going to eventually attach to these chromosomes to help split. Next, we have the metaphase. I always think of the metaphase as the middle phase because what's going to happen is those chromosomes that we formed in the prophase are going to be lined up in the middle. Those spindle fibers have now attached to the chromosomes, and what you're going to see is these centromeres are going to start to pull in opposite directions to split this chromosome in half and then have a full set of DNA on both sides. Next is the anaphase. In the anaphase, the chromosomes have split and they began to pull apart, and you can start to see two daughter cells starting to form here as those spindle fibers begin to pull in opposite directions. And then you have the telophase. And in the telophase, you're going to have the reformation of the nucleus. So this nucleus membrane here is going to start to reform. You can see the chromosomes starting to dissipate as we have started to fully form our two daughter cells. Okay. The final step here after telekinesis, it kind of goes hand in hand, is cytokinesis. And this is where the cells actually split. The cytoplasm is split by a cell membrane, and you have two fully formed cells. And these are going to be considered the daughter cells because they were made by the father cell, which was the original cell. Okay. All right, so that's going to be it for this one, real short lecture. We'll talk more about this interphase tomorrow in class. I think it's just a little better to discuss this in person, and then we'll work on making some models. That's going to be it for today, and I will see you all tomorrow.